So chapter 10, we're actually going to go through part of it today and then the other part on Monday. So we're going to split it up. There's a lot in chapter 10 and our brain is really complicated, so we're going to save that, most of that for on Monday. And then Wednesday we'll do the endocrine system, and then Friday we have a vacation, right? Next Friday we don't have class. Yeah. You got it. Oh, you have a race. No vacation for you. Sorry. Okay, here we go. So our nervous system is essentially made up of three things, and it's our brain, it's our spinal cord, and then all of the nerves that branch out from the spinal cord and go out to the rest of the body. And so the job of this system is kind of like the U.S. Postal Service, right? Okay, it's our messenger system. So this system receives messages, it transmits messages, so there's like stuff coming in, stuff going out. And along with the endocrine system, the nervous system coordinates all the activities of like all of our trillion cells in our body. So this is the boss, right? It tells everything what to do. So the medium for the endocrine system, however, is hormones. And we'll talk about that in chapter 12. But the medium for the nervous system is electricity. And so electricity travels through our nerves and then innervates. That's the word for when a nerve reaches its target, when it innervates and then sends that impulse into that organ. So the nervous system contains two main divisions. The central or CNS, which is basically the brain and the spinal cord. And then the second division is the peripheral nervous system, or the PNS, and that's everything else. So everything that goes off of the spinal cord. I'm going to draw kind of the branches of everything up here so that we can see it the whole time while we're talking. So we got the CNS. This is going to be the brain, and the spinal cord. And then we've got the PNS. And we're going to go through all these divisions as well. So let's dive a little deeper into these subdivisions of the PNS. So subdivision 1, if you look up here on the right side of the slide, you'll see you have an afferent division and an efferent division. And so I want you to think of your afferent division as your inbox. And so this is where messages are coming from the nerve endings, like out on the outside peripheral part of your body, and it's going up to your CNS. So it's sensory, it's coming in, right? It's your inbox. And then I want you to think of your efferent division as your outbox. So these are messages that travel from your CNS out to the rest of the body, so the other direction. So let's make these divisions right here. We've got afferent. This is your inbox. And then we've got efferent, which is your outbox. So now let's subdivide again, okay? Starting with that afferent and efferent. So within the afferent division, there are two additional divisions. One's called the somatic and one's called the visceral. And they're called sensory. And so the somatic sensory are messages that come from your skin, from your bones, from your joints, from your muscles, all of those things that make up your structure. And then your visceral sensory are messages that come from the gut organs. So your heart, your lungs, your stomach, your bladder, all those things kind of in your thoracic and abdominal pelvic cavities. So let's draw these divisions here. So you've got somatic sensory. So these are messages from structural organs. And then you've got visceral sensory. And these are messages from gut organs. Visceral meaning your viscera, kind of inside the gut, right? So now let's look at just the efferent division. So efferent division, there's also two subdivisions, and these are called somatic motor and autonomic motor. So somatic motor are the messages that allow voluntary control of your muscles, of your bones, all of that stuff that makes up your structure, again. And then autonomic motor are the messages that control your organ functions, those things that you don't have to think about, right? So all those, the gut organs down that we get the messages from in the visceral sensory. So let's, and these are going to be involuntary control. So let's divide this stuff out, okay? So we've got your somatic, or no, I'm sorry. It is your, somatic motor. These are going to be voluntary control of those structures or structural organs. And then we've got autonomic motor. This is going to be involuntary control of other organs. So something's telling your heart to beat, right? But you don't have to think about it. That's this autonomic motor. And then things and changes in your heart are being noticed, but you're also not thinking about it. And so that's going to be this visceral sensory. It's messages sent to the brain from those organs. Make sense? Now, when we talked about somatic motor in the last chapter with muscles, because those are voluntary movements. The brain is saying, I need you to move your arm, right? And so the signal is voluntary and it's sent to those structural organs like your muscles that then pull on the bones and you create movement. That's this, somatic motor, okay? And then say we were to touch something hot and we have to do something about it, like remove our hand, that's going to be somatic sensory, okay? We're actually thinking about it somewhat. There is some sort of response to that, and it's something coming from those structural organs. Guys, make sense, all the divisions? Okay. So now we're going to subdivide one more time, okay? And this division happens only within the autonomic motor system, so that bottom one down there. And so there's two divisions here, and they're split up between one that stimulates arousal and one that stimulates calming, okay? So arousal will be your sympathetic division of the autonomic motor, and then calming would be the parasympathetic division of the autonomic motor. So let me draw those out. Sympathetic means arousal. And parasympathetic means calming. An example here would be your heart rate again, okay? So autonomic motor is going to be the division that controls your heart rate. When you get excited or nervous or scared, your heart rate goes up, right? So that would be a response that coincides with the sympathetic division, arousal. It increases your heart rate, okay? Now when you go to sleep and your heart rate goes to like really, really low and you're resting, that's going to coincide with this parasympathetic division of the autonomic motor system. And so that's calming your heart rate down. So when you exercise, sympathetic takes over at a certain point. When you go to rest, parasympathetic takes over. Right now, most likely parasympathetic's doing its job. That's why it's hard to stay awake in class, right? All right. So now let's talk about the cells that are included in this system. You know, every cell or every body system has their specific cells. These are the nervous system cells. So there's two types that come in the nervous system. You've got neurons, and we'll talk about those in a second. And then you've got neuroglia, and we're going to talk about those right now. So neuroglia are also called glial cells. And so they're the supportive cells for our nervous system. And so they protect the neuron, they'll glue them together. They also perform various functions that enhance the performance of all of the neurons. And so there's approximately 50 glial cells supporting every single neuron. It's crazy, right? So we have a ton of these. And so for each neuron, let's talk about the different types of glial cells. So first of all, you'll see on this list, we've got oligodendrocytes. So these are found only in the central nervous system, okay? So CNS. And so these cells are what make up the myelin sheath that cover the brain and cover the spinal cord. We'll talk about myelin in a second as well. So oligodendrocytes create myelin. The next one on here, the ependymal cells, these are also found in the CNS, the central nervous system. And these cells provide the inner lining of things such as the spinal cord, as well as all the inner lining of the cavities of your brain. Kind of think about like the inner lining of an organ or something like that. The inner lining of of the organs in the central nervous system are made up of ependymal cells. So these cells are responsible for secreting cerebrospinal fluid, or CSF. We'll also talk about this in a second. This is very, very important to our central nervous system. So then the next one, you've got microglia, also found in the CNS. So microglia are kind of like your house cleaner. They're like the lysosomes of your organelles and your cells. So they chomp up all the debris, they get rid of the bad stuff, they clean up, right? They remove all the dust and dirt. And so this process that they go through when they're eating up all the bad stuff, it's called phagocytosis. So microglia perform phagocytosis. So then the next glial cell we have on here are astrocytes. And so these are also found in the CNS. So these first four are all CNS. And these cells attach to the neurons, or they attach neurons to blood vessels, okay? And so they help form the blood-brain barrier. So the barrier, this barrier exists to protect the brain from foreign substances. And so it only allows nutrients through and not blood, okay? So they're like this barrier between the very, very highly important brain cells and then our circulatory system. And so things in the circulatory system have to go through the astrocytes before they'll even make it into the brain. So blood doesn't go through there. There's actually no blood passing through to those cells, just the nutrients that those cells need. So then the last one you've got are Schwann cells. So Schwann cells are found in your peripheral nervous system, not the CNS. So these are PNS cells. But they're essentially the same thing as an oligodendrocyte. They make up that myelin sheath, but they make up the myelin sheath for all the neurons that are in the peripheral nervous system. Oligodendrocytes make up myelin sheath in the central nervous system. Schwann cells make up the myelin sheath in your peripheral nervous system. So let's talk about this blood-brain barrier for a second. So these astrocytes They're actually the most numerous of all of those glial cells that we just looked at. We have the most of astrocytes. And so they're pervasive around the brain, right? Because they make up this barrier. So they're shaped like a star, hence their name astro. And they have these little tiny feet, these little cupped feet that actually are at the ends of all the projections on the star. And this allows them to latch on to the capillaries that are near the brain. Okay, so they stick to it. And so they will take the stuff from those capillaries and they will feed the brain with it. Okay, so things that the brain needs are glucose, right? It needs other nutrients. It needs, glucose is one of the biggest things. However, it wants to prevent anything else from getting there that the brain doesn't need, okay? Because the brain is highly important, right? And so our body has created this whole mechanism that is like a stop, okay? And it's like, wait a second, you look foreign, you shouldn't be here, only glucose can get in, you can't. It's like going into the president, in the Oval Office of our president, right? Okay, you can't just like walk into the Oval Office. You got to go through like a series of checks, right? Are you allowed? Is it okay? You raising your hand? Yeah. Yes, oxygen, glucose. Yeah. You got it. So this is, it's interesting though because a lot of medications won't pass through those astrocytes because they're seen as foreign. And so this is why it's really hard for doctors to treat brain disease or certain types of brain diseases because of this barrier. It doesn't just infiltrate, like if you take a pill and it reaches, you know, your pain in your foot because it just is allowed to, right? You're like, things like that don't just happen through the brain. So certain medications, only certain medications can make it through. All right, so the most numerous of glial cells are what? Astrocytes, you got it. Good job. Okay. So now let's switch to the neurons. We talked about the glial cells. Now we're going to go to these other nerve cells called the neurons. And so it's basically everything else, right? So neurons are the messengers. So they handle all the communication. And so there's three types of neurons. You've got sensory, which you can also call afferent, because you can see on the peripheral nervous system, we've got the inbox, there it's sensory, it's afferent. And they detect stimuli such as like touch or pressure or heat or cold, and they transmit that information about that stimuli up to our central nervous system, okay? So it's going from the periphery, sending messages to the central nervous system. Then the next type of neuron we've got are interneurons, okay? So interneurons are like the switchboard, okay? So they connect the sensory ones with the motor ones, which we'll talk about in a second. So they provide this like communication between the two. And then you've got the motor neurons, also known as efferent, the outbox. So they relay messages from the CNS out to the periphery, okay? So afferent comes into the CNS, efferent goes out of the CNS, out to the periphery. So let's visualize this process, okay? So we've got our hand, right? We're about to touch a hot pan, okay? So when it happens, there's sensory organs in your hand that sense that heat, right? And so this message is going to get sent through which neuron? Tell me. Or afferent or efferent? Sorry. But yes, you got it. Yeah, afferent. Good job. So it's going to be sent through, this message is going to be sent through an afferent neuron up to your central nervous system. It's going to start with your spinal cord, okay? Now, the interneuron is going to receive that message, and then with the brain is going to make a decision about how we want to respond to this heat, okay? then that decision that's made in our higher part, our central nervous system, gets sent out to our hand through the efferent motor neuron, and then we move our hand. And this happens like speed of lightning, right? Thank goodness. It doesn't happen as slow as it took me to talk about it. It actually happens like that, okay? So it's good. So the structure of a neuron should be a review to you, but let's take a look at it again. You've got your cell body, or the soma. It's up at the top there. This is where the nucleus resides. And then you've got the dendrites around that cell body, and they receive the impulse from surrounding cell bodies, from other neurons. And then you've got the axon, and this is where the impulse leaves that cell body and heads out to the very end. So now let's add a few more structures that we haven't talked about. So the axons of many neurons, not all neurons, but many of them, are encased in this stuff called myelin. It's called a myelin sheath. And this insulates the axon. And so the myelin sheath, which actually consists mostly of fats, of lipids, is formed by Schwann cells, right, in the peripheral nervous system, and then oligodendrocytes in the central nervous system. And so they're kind of arranged in these like sausage link looking places. You can see that on the slide there. And then there's these spaces between each of these sections of the myelin sheath, and those spaces are called the nodes of Ranvier. And so they're evenly spaced between each other. And then at the very end of the axon, you'll see kind of that broccoli looking thing. And this is your synaptic knob. And so this is where the terminal of the axon is, and which holds the vesicles of the neurotransmitters. Okay? You guys remember the neurotransmitter for a motor neuron? You guys just did it on your test. You can give me the abbreviation? ACH. You guys got it. Yeah. So ACH would be in little vesicles or little pockets in these little synaptic knobs down here at the end of the axon. So not all neurons are myelinated though. So myelin is actually beneficial because it speeds up impulses. And so the electrical impulse travels faster in myelinated neurons than it does non-myelinated neurons. So examples of neurons stimulating the secretion of, say, your stomach acid are not myelinated because speed isn't a big deal, okay? However, the neurons that are innervating your muscles for movement, those are myelinated because you want that to happen fast.
So what if you get an injury and you didn't notice? What causes that?
An injury what?
Like say you like cut your finger but you didn't notice for a little bit and then you're like, oh, my finger's cut. Like why does that happen? And then you go into shock and then that's when you start feeling it.
Oh, like in like a traumatic, like in big trauma? You know, I don't really understand how that happens, but I think, yeah, I think there's something going on up here in your central nervous system that's kind of prioritizing your focus on, like, say you need to, whatever the situation that caused the injury was dangerous, and all of a sudden you are focused mostly on getting away from whatever the danger is. And then you get down the road, you're out of danger, and all of a sudden you're like, oh my God, my leg's broken, or my foot is bleeding, or whatever. Yeah, and I think it's just kind of like a fight or flight response that makes your body go, what's the biggest priority here? Let's focus on that, and then all of a sudden you're like, oh no, I'm bleeding. Yeah, our brain is crazy. Our brain is, we don't even understand our brain. Our brain doesn't understand our brain. All right. So nerves that stimulate those muscle cells, like I said, are myelinated because we want that impulse to travel fast. We're looking for speed here. So those that are myelinated in the peripheral nervous system, who can tell me what is the cell that creates the myelin in the peripheral nervous system? Good. And then the CNS, it's the oligodendrocyte. Good. You got it. Okay. So Schwann cells, let's look at the peripheral nervous system here on the left. So the Schwann cells will actually wrap themselves around the axon, like layers, and they'll lay down multiple layers of the cell membrane. And so the inside layers form that myelin sheath, but then the nucleus and most of the cytoplasm of this Schwann cell are actually on the outermost layer. Okay, so it's kind of like it's squeezing all the gooey stuff to the outside of it where the nucleus and the cytoplasm are. And so the outer layer of that is called the neurolemma. So like we have the sarcolemma on a muscle fiber, it's a neurolemma on a neuron. And so this is essential for an injured nerve to regenerate. That neurolemma with the nucleus there and that cytoplasm and all the other organelles right there, okay? However, in the central nervous system, if you look over to the right, the projections from one oligodendrocyte will wrap around multiple axons of several neurons around them, okay? So then that means that the nucleus and the cytoplasm and all of the organelles aren't necessarily attached to all the neurons that the sheath is wrapping around. And so therefore there's no neurolemma wrapping around those specific axons. So this is why when you have an injury to your brain or your spinal cord, it's considered permanent, right? Because they don't regenerate. You need that neurolemma to regenerate. But on peripheral nerves, you can actually regenerate that because that neurolemma, where that Schwann cell is positioned around that axon, allows that nerve to repair itself. That's why it's a big deal, right? You know, when you injure your spine or your brain, people become paralyzed. You know, some of those injuries become permanent and can change people's personalities even, right? So an interesting fact is this myelination happens, it actually starts during the 14th week of fetal development. So when you were in your mother's womb at 14 weeks, you started developing your myelin sheath. And it will actually last, this development will keep going all the way through adolescence. So it's a really, really long period of this development of myelin. And so your mother's nutrition, what she eats, her health, is absolutely impactful on this myelin development. And then of course your health as a child through adolescence is also impactful. Has anyone heard of multiple sclerosis, MS? So this is a disease that is essentially causing this myelin sheath to break down. And so their impulses can either be too great and they can have spasticity, or they will start to lose innervation to certain parts of their body. And so somebody with really, really advanced MS will eventually lose the ability to walk and be able to use their hands correctly. And it's all because of this degradation of this myelin sheath around the neurons. So which neuron detects sensation, such as touch or heat, and relays information about that stimuli to the CNS? C, afferent, sensory. You got it. Good job. All right. So now let's talk about how that neuron transmits its electricity. So this is called an electrical impulse, okay? So at the molecular level of electrical transmission, it's basically an exchange of ions across a membrane, okay? And we're gonna get kind of chemical when we talk about how a neuron sends electricity, but think about it, it's about ions changing position. So in the body, we've got oppositely charged ions, some positive, some negative, and they'll travel into and out of our cells across that membrane. And so whenever one side of a membrane, say the inside or the outside of a cell, doesn't matter, if one has one charge and then the other side has the other charge, that's called potential, okay? And so the potential exists for them to move towards one another. You could also call it membrane potential, and they're on opposite sides. So then when there's an excess of positive ions on one side and an excess of negative ions on the other, the membrane is said to be polarized, okay? So if you've got negative on one side, positive on the other, it's polar, right? It's opposites. And so if it's an excess on either side, it's going to be polarized. So now, when a neuron is at rest, meaning that it's not conducting any type of electrical signal, the interior is going to have a negative charge, okay, a negative electrical charge. And then the exterior, on the outside of the membrane, is going to be a positive electrical charge. So the interior will also contain an abundance of potassium ions, okay? And then the exterior is going to be rich with sodium ions. So right now, in resting potential, you've got negative on the inside of the membrane, positive on the outside of the membrane, a lot of potassium in here, and a lot of sodium out here. However, potassium isn't negatively charged though. It's positively charged. Both sodium and potassium are positively charged. So what makes up this negative charge are tons of other ions in there that are very negatively charged, and then also some nucleic acid. So this negative and positive is going to be separate from the potassium and sodium, okay? So because of the membrane's permeability, because things can pass in and out of it, a certain amount of sodium and potassium ions will leak across the membrane. However, there is the sodium-potassium pump. You guys remember that from, I think it was chapter 2, 3, I don't remember. That's always working, constantly working to make sure that they're in the same place or in the right place, right? And so who can remember the sodium-potassium pump? Well, what goes out and what comes in? And how many? Yeah, 3 sodium out, 2 potassium in. And it costs an ATP every time that happens, right? So let me write that out just so you guys can remember it. Sodium, potassium, pump. So this is active, right? Uses ATP. We've got 3 sodium out, 2 potassium in. Sodium is notated as Na+, potassium is K+. So this state of being inactive that this neuron's in right now, but also being polarized, where you've got a ton of positive on the outside, ton of negative on the inside, it's called resting potential. And so this means it's resting, but it has this potential to react if a stimulus comes along. So the sodium-potassium pump is working constantly to make this neuron stay in resting potential. So now let's say a stimulus does come along, okay? So it's going to come from this direction, right? Going to go into the dendrites, into the cell body, the stimulus from something else. So it could be chemicals, it could be heat, could be mechanical pressure, whatever it is. So this is going to create channels on the neuron's membrane to open, or cause them to open, and this is going to allow all that sodium that was on the outside to start rushing into the neuron, okay? So there's going to be a channel, there's a bunch of channels all along this membrane that allow sodium to rush in. And this is going to be a passive movement. No ATP is used, no sodium-potassium pump is used. Just the stimulus comes in and opens these sodium channels. And just like, you know, if you've got a lot of pressure out here of the sodium, it's going to flush in, right? So now you've got all this sodium starting to come in. So now these channels are different, right, than the sodium-potassium pump. And so it's passive, just letting the sodium in. And what this does is it adds a bunch of positively charged ions to the inside of the neuron now, right? And so this changes the charge of this region of the cell. So if it starts right here and sodium is just rushing in, now this part of the neuron is going to start becoming positive. And then the outside is going to start becoming negative. Okay? So this is this next step. And so when this happens, the membrane is said to be depolarized. We're no longer polarized, right? Because we're starting to have this change, and we're starting to have a difference in the charges on the inside and the outside. So remember that there was that all or nothing rule for motor neurons, right? And so this goes for all neurons. It has to reach a threshold. And so this depolarization has to reach that threshold level before any other signal is sent. But once it does, adjacent channels open up and allow even more sodium in. So we started right here, right? And now it's reached this threshold and it starts to just like go boom, 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 all the way down, just allowing more sodium in as it's going down the axon, okay? And so when excess positive now goes flushing into the inside and the outside is negative, this is called action potential. This is when that impulse is being sent down that axon, okay? So sodium channels are starting to open one after another as it's going down the axon. So the action potential will continue down the axon as one segment stimulates the next segment that stimulates the next segment, okay? Make sure I don't want to cover anything else. So now all of this sudden influx of sodium triggers other channels to open up that send potassium out, okay? So it's trying to balance itself out. So just like on the last slide, we had sodium channels letting all the sodium in. Behind this process, we've got now channels that are pushing potassium out, okay? So now we're getting a big flip-flop in charge and we're getting a big flip-flop in our ion concentrations. So sodium goes in, goes down the axon, and then follows that, or what follows that are potassium channels that allow all that potassium to go out. So this actually repolarizes the cell, okay? So we're not depolarized anymore, we're repolarized. But it's flip-flopped. And so this is what is we're not going to be able to do another impulse until this gets changed back to its original resting potential. So it'll become repolarized, like repolarization here, but it's not going to be set up for an impulse. So as long as that sodium and the potassium are on those opposite sides of the membrane, the neuron won't respond to a new stimulus, even though that membrane is polarized. And so this is called the refractory period. So this neuron wants to be in its resting potential state, right? It wants to get back to the beginning. And so now we've got the sodium-potassium pump that comes into play. And so it works to return all that sodium back to the outside and bring all of that potassium back in. And when this is complete, the nerve is again polarized and then is in resting potential and it's ready to receive another stimulus. So we've got kind of three things happening in this membrane, right? We've got sodium channels that are passive. We've got potassium channels that are passive. And then we've got the sodium-potassium pump that's active using ATP to restore everything back to its happy place. Make sense? So when this is going on, this is called the refractory period. You guys want to watch the video on it, an animation? Walk through the whole thing? Let's do it.
To relay messages to organs and tissues throughout the body, nerves must initiate and then transmit signals from one neuron to the next neuron at lightning speed. Signal transmission occurs through an electrical current. When a neuron is not conducting an electrical signal, its interior has a negative electrical charge, whereas the charge on the outside is positive. The outside of the cell is rich with sodium ions, whereas the inside contains an abundance of potassium ions. The interior of the cell also contains large, negatively charged proteins and nucleic acids. These additional particles give the cell's interior its overall negative charge. Because of the membrane's permeability, a certain amount of sodium and potassium ions leak across the membrane. However, the sodium-potassium pump constantly works to restore the ions to the appropriate side. This state of being inactive and polarized is called resting potential. The neuron is resting, but it has the potential to react if a stimulus comes along. When a stimulus comes along, channels on the resting neuron's membrane open, and the sodium from outside the membrane rushes into the cell. The addition of all these positively charged ions changes the charge of a region of the cell's interior from negative to positive. As the membrane becomes more positive, it is said to depolarize. If the stimulus is strong enough, adjacent channels also open, allowing even more sodium to flood the cell's interior. This creates an action potential, meaning that the neuron has become active as it conducts an impulse along the axon. The sudden influx of sodium triggers the opening of other channels to allow potassium to flow out of the cell. Soon after potassium begins to exit, the sodium channels shut to prevent any more sodium from flowing into the cell. This repolarizes the cell. However, the outside now contains more potassium, and the inside contains more sodium. Although the membrane is polarized, the neuron will not respond to a new stimulus as long as the sodium and potassium are on the wrong sides of the membrane. The sodium-potassium pump works to return sodium to the outside and potassium to the inside. This is known as the refractory period. When the sodium-potassium pump restores sodium ions to the outside of the cell and potassium ions to the inside of the cell, the nerve is again polarized. It remains in resting potential until it receives another stimulus.
All right. Make sense? Yes.
So in depolarization, is it a 1-to-1 ratio, and then in the refractory period, is that 2-to-3, or is it the same ion all?
I don't think it's a 1-to-1. I think it's a lot of both. Yeah. The channels are just actively letting them through. It's not just like one going in, one going out. It's like kind of like an influx. The sodium-potassium pump, however, is active, and therefore it's very specific. And it has those sites within the pump that only allow that many ions to attach. Good question. Any other questions on that? Make sense? Cool. All right. So that was basically what it would look like if there was no myelin on the neuron, right? On the axon. And so myelinated nerves actually work a little bit differently. And so the myelin actually blocks the free movement of those ions from going across the membrane. And so the only place that those ions can exchange across the membrane are in between those myelin sheaths, so the nodes of Ranvier, okay? So those little spaces in between each of those little sausage links. And so the action potential or electrical charge, the change actually occurs right there. And so the current, the flow of electricity runs underneath the myelin, where it triggers another action potential at that nodes of Ranvier. So it's instead of it happening like right here, right here, right here, right here, right here, right here, it goes boom, 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 boom. So now you can kind of see how this might speed up the process, right? So if there's no channels here, it's like this impulse will leap from one node of Ranvier to the next. Okay, so instead of it happening like every whatever, you know, I don't know the distance, but along the membrane, it goes boom, 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 boom. It's a lot faster to hop to the wall from where I'm standing than to step to the wall from where I'm standing. Does that make sense? So the impulse is going to travel faster when there's myelin because of those leaps from the nodes of Ranvier to the next nodes of Ranvier. So what is another term for a nerve impulse? So when it's actually sending an impulse down the axon. Guys are guessing and whispering. D. So think about this. If the nerve impulse is being sent, it's in action, right? And so that's going to coincide with action potential. The rest of them are these states of before and states of after. The action potential is when that impulse is going right down the axon. Okay. So let's keep this message going, right? It's gone down the axon and it's about to be sent to another neuron. And so it has to pass through this place where that synaptic knob touches the dendrite of another neuron, right? And that's called the synapse. So here's how this whole process works. So when that action potential reaches the synaptic knob of that presynaptic neuron, so I would call this one the pre, and this one's the post. So when it reaches that synaptic knob of the pre, the membrane depolarizes, right? And so the ion channels open up. And then now we've got another ion that comes into play. We've got calcium. We've been talking about sodium and potassium. But now when we get down to this synaptic knob, these channels will open up and calcium flows into the cell. So this infusion of calcium is what's going to cause these vesicles that have those neurotransmitters in them to bind to the cell wall right here, and then they open up and release their particles out into this area, the synaptic cleft. So these hang out here until they get some sort of signal, and that signal is this calcium fluxing in. And that calcium doesn't flux in unless there's an action potential. Got it? So now we've got this neurotransmitter. It's all these little blue dots here. And they're released into this area, and then they have their very specific receptor on the postsynaptic neuron. Now this neurotransmitter is going to say one of two things. It's going to say an excitatory thing or an inhibitory thing. It's going to say send that impulse down or don't do anything, okay? So what happens if the neurotransmitter is excitatory? The impulse is going to cause sodium channels to open up on the postsynaptic neuron, okay? So sodium's going to start flowing in if it's excitatory. Now, if that neurotransmitter is saying that it's inhibitory, potassium channels are going to open and nothing else is going to happen, okay? So potassium is associated with inhibitory from those neurotransmitters, and sodium is associated with excitatory from those neurotransmitters. Now, once this whole process is done, the receptors are going to release that neurotransmitter back into the cleft, and either an enzyme, you guys know acetylcholinesterase, or I said that wrong, but you know what I'm saying, ACHase, eats up the remaining neurotransmitters, or they can actually go back and be recycled into vesicles again. And so this is where that process happens. So this is actually another really awesome video from the beginning of what we just talked about with membrane potential all the way through the synapse. I'm going to let you guys watch this on your own because it's kind of long, but it's embedded in this slide so you guys can see this. It's a really good video. All right, so let's transition now. Let's think about the central nervous system. And we're going to start with the spinal cord. So the spinal cord relays messages from the brain, right, out to the body. And then it extends down the base of the brain all the way to the first lumbar vertebrae. So the cord begins to separate there at the lumbar vertebrae into thinner nerve roots. And this little area is called a cauda equina. And so it gets its name because it resembles a horse's tail. So caudal meaning rear and equine meaning horse. And so you get this little branching of nerves at the lumbar vertebrae, and it's called the cauda equina. So now branching off of the side of the spinal cord, we have 31 pairs of spinal nerves. And so they're divided into regions that coincide with all of the vertebrae, and you guys are familiar with these regions because you learned the vertebrae, right? And so nerves from your cervical region, you can see up there, they're going to innervate your chest, your head, your neck, your shoulders, your arms, and then down to your hands. So all that stuff that kind of just comes out from right there, right? It's also going to innervate your diaphragm. So your diaphragm's innervated by the cervical region of those spinal nerves. Next, the nerves that come from the thoracic region, that go from T1 to T12, they're going to extend to the intercostal muscles of the rib cage, so it helps you with breathing. They're also going to innervate the abdominal muscles and then the muscles of your back. So everything that's in that region, right? And then your lumbar spinal nerves are going to innervate the lower abdominal walls and then parts of your thighs and then your legs as well. And then the nerves that come from your sacral region will extend down the thighs, through your buttocks, the muscles in the skin of your legs and your feet, and then also the anal and genital regions. So you guys remember what a vertebrae looks like, right? You got the spinous process that's on the posterior side. You got the vertebral body that's on the anterior side. That's where the disc sits, and then the next vertebrae sits, and then the next disc, and so on, right? So the spinal cord sits inside that open column that runs down your vertebrae. So you got the body, and you got the spinous process, and then there's this big foramen, spinal foramen, and that's where the spinal cord runs. And so this is a cross-sectional view. So if you were to take the vertebrae and flip it up and look at it cross-sectionally, that's what we're looking at right here. So in your spinal cord, you actually have two types of tissue. You've got white matter and gray matter. And we're gonna talk about these types of tissue in our brain as well. So gray matter appears gray because it doesn't have any myelin. And it mostly contains the cell bodies of the motor neurons, so where those nuclei are, right? And the H-shaped mass is divided into two sets of horns. So this gray matter right here, where most of the cell bodies are, it's got two horns. You've got a posterior side and an anterior side. And you know because this vertebral body is anterior on the vertebrae and the spinous process is posterior on the vertebrae. So now white matter appears white because it's got a ton of myelin. So gray matter doesn't have any myelin, full of cell bodies. White matter has a bunch of myelin, and therefore it's a bunch of axons. And so these axons run down this white matter in what's called tracts. And we're going to talk about tracts in a second. And so they carry all those impulses from one part of the nervous system to our next. And then there's this tiny little canal. You can barely see it. It's basically like a dot on the slide in the middle of the gray matter. It's called your central canal. So that cerebrospinal fluid flows down through that central canal. And then there's actually an epidural space. And it's mostly made up of connective tissue and adipose tissue. And it's what pads the spinal cord around the vertebrae. So if you were to get like a shot in your back called an epidural, they're sticking a shot right into that space so that you're going to get numb, right? Women that don't want to deal with any more pain from pregnancy will get an epidural, and it just blocks the feeling of all of their body from that part down. So remember that the vertebrae are each connected to each other by that intervertebral disc, right? That padding in between the vertebral bodies. And so it's going to provide a gap between the vertebrae. And so within that gap is where those spinal nerves are going to leave the spinal cord and go out to the left and the right side. So they squeeze out that little gap there. And so each gap is going to have a dorsal side and a ventral side. And so dorsal means posterior, ventral means anterior, right? So you can see the two there. And so these two roots are basically your inbox and your outbox into and out of the spinal cord. So the dorsal root or the posterior root will carry information into the spinal cord, and the anterior root will carry information out of the spinal cord. So within the dorsal root also you can see this kind of like bump right here. It's called the ganglion. It's just a collection of a bunch of cell bodies in there, and it just makes like a big bump. So these two roots will join eventually, and then they'll contain mixed neurons. They'll have neurons that go both directions once they join. And so they'll contain sensory and they'll contain motor. And so this fusion of those two roots, the dorsal and the ventral root, is actually what's called the spinal nerve. At this point it's called the spinal nerve. And so protecting the spinal cord is the vertebrae, of course, the bone. But there's also some layers of fibrous connective tissue called meninges. You guys ever heard of meningitis? Yeah, it's an infection of these layers. So there's 3 layers. And so meninges is the term for all of them, but you'll see them identified as motors when you're talking about them specifically. So the most inner one you'll see is the pia mater, and it's going to wrap right around that spinal cord. And then the next one is going to be the arachnoid mater, and this is going to be kind of like a web-like shape to it. And so arachnoid is another word for spider or spider. You know, it's like you guys know what arachnophobia is, like the fear of spiders. So that word means it looks kind of like a web, right? And then there's a space in between the pia and the arachnoid called the subarachnoid space. And then you've got the dura mater. The dura mater is going to be that most outer layer of the meninges. So now within the spinal cord, there's specific routes that these impulses will travel. And these routes are called tracts. And so they lie within that white matter, and they're made up of a bunch of super, super long axons, like tons of long axons. And you've got ascending tracts that convey sensory information, and then you've got descending tracts that relay motor information. And so all of the nerve fibers within a single tract will have a similar origination, destination, and function. So they're kind of like, if you think about a track as like a private road that only specific cars can drive on. And so it's a route, it goes from point A to point B, but only very specific vehicles are allowed. And so you've got tracks that allow each of those specific vehicles. So here's some of your most important ones right here. The dorsal column relays sensations of deep pressure, vibration, proprioception. It's basically your body awareness. Then you've got the spinocerebellar tract that's responsible also for proprioception. Got the spinal filament. Oh my God, I'm gonna butcher these names. Spinothalamic tract that relays sensations of temperature, pressure, pain, and touch. And then you've got some descending tracts, and those are the corticospinal, which are responsible for your fine movements of your hands and your fingers and your feet and your toes. And then you also got the pyramidal tract. And you actually have an extrapyramidal tract as well, and those are both associated with balance and muscle tone. So there's actually identical tracts on either side of the spinal cord. It doesn't show that here, but just know that you've got stuff that goes to and from the right side of your body, stuff that goes to and from your left side of your body, okay? So the left side of your spine is going to coincide with the left side of your body. The right side of your spine or spinal cord coincides with the right side of the body. However, this is weird, it switches in your brainstem, right? So all the information from the right side of your body switches places back here in the brainstem and then gets processed on the left side of the brain. And then vice versa for the other side. So what is actually a nerve? Okay, so that word nerve, we're talking about neurons, right? So what's a nerve then? And so a nerve is essentially a bundle of neurons. Mainly they're axons. And so you can also hear them called nerve fibers. So a number of nerve fibers contain a single nerve, varies from just a few to approximately 1 million. So you can have one nerve fiber or neuron in a nerve, or you can have millions of nerve fiber neurons in a nerve. So kind of like our muscles, they tear down from like big to small, right? And so you've got also bundles of nerves, and they're called fascicles, just like bundles of muscle fibers are called fascicles. So the fascicles are individually wrapped in dense connective tissue, and it'll actually contain blood vessels here. So most of the time, these nerves will contain both sensory and motor, so both afferent and efferent, and they're called mixed nerves. However, some of them are specific and are only sensory, and then some are specific to only be motor. But most of the nerves in your body are going to be mixed. Most of them have both motor and sensory. Just like your spinal cord regions and your vertebral regions all were the same, the spinal nerves are going to be the same. They're going to coincide with all of those regions. You've got 8 cervical, because one spinal nerve actually leaves above the axis, C1. You've got 12 thoracic, 5 lumbar, 5 sacral, and 1 cossageal. So they're symmetrical, right? You've got one that departs from the left and one that departs from the right. And so outside of the spinal column, sometimes these nerves will form networks, and they're called plexuses. And so the 4 major plexuses in your body, you've got a cervical plexus, you've got a brachial plexus, you've got a lumbar plexus, and a sacral plexus. So you can see them where they are, and they coincide with those colors. So the cervical plexus is in green, brachial plexus is in orange, lumbar is purple, and sacral is blue. You can see where they all go. So there's actually locations on your skin that coincide with these plexuses, and they're called dermatomes. And so you can actually touch your skin, or somewhat like a doctor can touch or stimulate a place on your skin and see if that plexus is actually working or not. And that's what those dermatomes are. And I think that's in your book. I think it's page 175. You can see where all the dermatomes are. So now sometimes our body will sense things and respond to it and totally bypass our brain, okay? And we won't even think about it. That's essentially what a reflex is. We'll sense something, it'll respond with the motor some sort of action, and it won't even make it up to our brain. It won't even go up to the higher command center. And so, for instance, say you touch something hot. This is actually where a reflex happens. So that information is going to go straight to the spinal cord, and it's going to actually reflex, called a reflex arc, from a sensory neuron to a motor neuron, and you immediately pull your hand back. You don't think about it, right? Sometimes it just happens like that. That's what a reflex is. So another example would be our patellar flex. Who's been to the doctor and they've hit the hammer on the front of your knee, and all of a sudden your leg just shoots up and you're like, whoa, how'd that happen? That's another example of a reflex. So in this instance, the doctor hits that patellar tendon, and it will actually pull the muscle slightly, and there's a receptor in here that senses that pull. It sends the message to the spinal cord. It arcs in a reflex arc right here and goes straight back out as a motor response, and your leg goes bloop, like that. So it doesn't go all the way to the brain. It just flips sides in the spinal cord. Question, bundles of axons within the white matter that serve as routes of communication, those private roads, what are they called? Good, tracks. Good. All right. How you guys doing? Fading yet? A little bit? I could stop here, or I could go a little bit into the brain, and we could pick up on Monday. There's still a lot to cover. What would you guys like? 1, 2, 3, 4. I got 4 more slides. Let's do it. Let's get through it. Stand up. Wake up. Let's get through it, because we got a lot still to do on Monday. Okay. So while you're moving around, I'm going to keep going, all right? Let's talk about the brain. So scientists and doctors are relentlessly studying the brain, right? Like what we know now about our brain, we didn't know last year. It's a very, very evolving science. Neuroscience is nutso. So if you guys really want to get into some crazy stuff, you could study that in grad school or something. But we still don't understand everything, right? There's so much going on up there. Our brain doesn't understand our brain. But there are some things that we do know, and we'll talk about them right now. Let's talk about these regions of our brain. So our brain is divided into 4 major regions. So we've got the cerebrum. It's going to be your outermost part, that wormy looking stuff. We've got the diencephalon. That's going to sit inside of the cerebrum. You can't really see it. It's just going to be kind of like the colored pictures in there. Then you've got the brain stem that's going to come out of that diencephalon. That becomes your spinal cord. And then you've got your cerebellum, a stripedy looking thing on the lower back side of the brain. So the cerebrum is the largest portion, and it's made up of thick ridges. The ridges are called gyri. And in between each of those ridges, you've got these little depressions or little like cuts. Those are called sulci, okay? So a gyri would be this raised part, and then the sulci would be the valleys in between the raised part. And so some of the sulci are very prominent and very deep, and those are actually called fissures. Now the cerebellum is the second largest part. You'll see that in the back lower side, and it contains more neurons than the rest of the brain. There's tons and tons of neurons in your cerebellum. And then you've got the brain stem, and it consists of 3 parts. You've got the midbrain, the pons, and the medulla oblongata. And then there's a deep groove that's not shown on this slide, but it runs in the same path as your sagittal fissure on your skull. Remember that one? This is called the longitudinal fissure of your brain. It's one giant sulci that goes from the front to the back. And it actually splits the whole brain into a right and a left hemisphere. And that's where you divide the sensations, or the control of your right side goes to the left hemisphere on the left side of that longitudinal fissure. And then the left side of your body gets controlled and sensed by the right side of your brain on the right side of that longitudinal fissure. So like the spinal cord, the brain also contains gray and white matter. However, it's flip-flopped. In the spinal cord, the gray matter is more internal and the white matter is more external. In the brain, the white matter is more internal and the gray matter is more external. And this gray matter that makes up the external part is called the cortex. And the cortex is responsible for your personality, your thoughts, your reasoning, your critical thinking. Your learning right now is being, your cortex is being used for that. And so these folds, all these gyri and these sulci, are basically increasing the surface area of that tissue so that you've got more space for more neurons. Now this will be a review. Just like your spinal cord has meninges, your brain has the same meninges. So you've got your, and they protect your brain as well. So you've got your cranium, right? Your skull, and that protects your brain. And then you've got the next meninge is going to be the dura mater. It's got two layers. One kind of connects to the inside part of that bone. And then the next one, you'll see it will dive down into the longitudinal fissure here. And you've got this space called the dural sinus. And then you've got the arachnoid mater. Same thing on the spinal cord, and then you got the pia mater. So pia is most internal, arachnoid's the middle one, and dura is the outer one. Now, cerebral spinal fluid is inside of the dural sinus and down into those fissures. And so, you guys, anyone ever had a concussion? Yeah. So, our skull and this fluid that our brain is floating in protects us from minor bumps and movements, right? But if something like an impact that's greater happens, then your brain will literally slam against your cranium, and that cerebral spinal fluid can't help you. And it's really crazy, right? Because the cortex is what's getting hit, right? It doesn't mean that it's not injuring other parts. This is why concussions, and especially multiple concussions, can actually change someone's personality completely. Has anyone ever had someone in their life that has gone through really severe multiple concussions? I had a friend that lost their sense of smell. Can't smell anything from a concussion. Yeah. What was that?
It doesn't taste good eating anything, right?
I would think so. Such a big part of it. Yeah, I mean, his taste is diminished, obviously. Yeah. So your brain is important, but it's fragile. It's very, very fragile. So protect your head. Walk around with the helmet on all the time. Just kidding. You don't have to do that. Let's talk a little bit about that cerebral spinal fluid. So kind of like our blood is constantly flowing through our blood vessels, cerebral spinal fluid's constantly flowing through our central nervous system. So it bathes the outside of the brain. It also flows through these four chambers, also known as ventricles, that are inside of the brain. So the cerebral spinal fluid is what's actually responsible for taking that oxygen and glucose that comes from those astrocytes and delivering it out to the rest of the brain tissue. So here are the four ventricles. You've got two lateral ventricles that arc through the cerebral hemispheres, one on the right, one on the left there. Then there's a space in the middle, and that's where the third ventricle is. You can see it in green. And then down lower, which is actually a long canal, is the fourth ventricle. And this ventricle will extend all the way down through that central canal that goes through the middle of your spinal cord. So where does this cerebral spinal fluid come from? So we have the ability to produce more CSF all the time. And it's done in these places within the ventricles called choroid plexuses. So the cerebral spinal fluid is not only important for the nutrients and the waste transportation, but it's also that cushion that cushions your brain in times of impact, right? So cerebral spinal fluid also monitors CO2 levels. And so your respiratory rate is actually going to respond to what the CO2 levels that are in the CSF. It's kind of like the messenger of what's going on in the rest of the body. So let's talk about this production. So the choroid plexus will start, where it starts is in the lateral ventricles. And so it starts to secrete cerebral spinal fluid up there. You can see number 1. And then that fluid will flow into that third ventricle that's in the middle there. And the choroid plexus that's there will add more fluid to the mix. Then it flows to the fourth ventricle where more CSF is added from the fourth ventricle choroid plexus. And then some of the cerebral spinal fluid will flow through 2 tiny openings called foramina, and it will make its way into that subarachnoid space, and it is able to travel up into the rest of the brain, okay? And at this point, it gets soaks in, and that's how you get all those nutrients to all that brain tissue that's outside of the ventricles. And then finally, the cerebral spinal fluid is reabsorbed into the venous bloodstream from the projections of the arachnoid matter that comes from those astrocytes. And those projections are called arachnoid villi. Okay. Last question here. Which statement about gray matter is true? Think about in your brain. What part of your brain is gray matter? Yep. So the cortex of your brain is gray matter, and the cortex is responsible for all your higher thinking, your thought, your reasoning, your personality, all that stuff. Good job. Okay, now I'm done. So, the Crash Course A&P actually has some awesome modules. And so my suggestion is to review them this weekend just for a little repeating of the information. Finish reading chapter 10. We're going to go into more on these divisions of the nervous system. And I will hopefully have this video posted at some point today. If you guys are experts at that stuff, come chat with me. Okay, have a good weekend.